Good morning. Good morning to everyone. Welcome to this session of today. We start with a talk from Professor Ian Gout. The title of his talk is The Regulation of Metastasis Suppressor NMA1 by a key metabolic cofactor coenzyme A. Please, it's a pleasure.
Thank you, Matilda. It is a great pleasure to talk at this conference, and I would like to thank Ira Skvortsova for the invitation and the opportunity to present our recent findings on the regulation of metastasis suppressor protein NME1 by a key metabolic cofactor coenzyme A. So to start with, I will introduce the topic of reactive oxygen species and the antioxidant response in mammalian cells. Then I will tell you about the role of coenzyme A in cellular metabolism and our recent discoveries of the antioxidant function of coenzyme A and a novel post-translational modification which we termed protein coagulation. And by the end of my talk, I will focus on covalent and non-covalent mode of regulation of metastasis suppressor protein NME1 by CoA in cellular response to oxidative and metabolic stress. It is well known that reactive oxygen species are constantly produced in mammalian cells, and they include superoxide, hydrogen peroxide, hydroxyl radical, nitric oxide, and others. They are mainly produced in mammalian cells in mitochondria, peroxisomes, and by several enzymes, including lipoxygenase, NADPH oxidases, cytochrome P450. It is also known that numerous exogenous factors can induce the production of reactive oxygen species in mammalian cells, including UV, ionizing radiation, chemodrugs, pro-inflammatory cytokines, and various environmental toxins. To cope with the significant increase of reactive oxygen species, mammalian cells devised two important antioxidant defenses. The first one is the expression of antioxidant enzymes, and these include catalases, superoxide dismutases, glutathione peroxidases, and others. In addition to that, low molecular weight cells are involved in the antioxidant response, and they include glutathione, and now we know coenzyme A, as well as vitamins A, C, and E. The level of reactive oxygen species in mammalian cells is tightly controlled because redox homeostasis is critical for essential cellular functions such as metabolism, the regulation of cell growth, the accumulation of mass, proliferation, and differentiation. It is now established that reduced level of ROS production can impair important physiological functions such as proliferation and the response to viral and bacterial infection. In contrast, when the level of ROS is produced for a long period of time, it can impair important physiological functions and can induce random cellular damage or damage to specific signaling molecules or pathways. This damage can result in cell death, various pathologies, and aging. Reactive oxygen species in mammalian cells are constantly produced at the basal level. However, when there are some specific regulatory signaling or metabolic processes, there is a temporary and localized increase of reactive oxygen species. And one example of that is signaling via receptor tyrosine kinases. So now it is well known that the binding of a ligand to a receptor induces receptor tyrosine kinase induces the activation of NADPH oxidases at the plasma membrane. They produce hydrogen peroxide, hydrogen peroxide, and hydrogen peroxide would inhibit receptor tyrosine phosphatases. And in these ways, it is a very good way to facilitate efficient receptor tyrosine kinase signaling. In contrast, when reactive oxygen species are produced for a prolonged period of time at a high level, it can result in oxidative stress. And cellular macromolecules, especially DNA, lipids, and proteins, can be damaged. Today, I will talk mainly about the regulation of cellular antioxidant response via protein coagulation and the regulation of CoA binding to proteins, focusing on NME1. Coenzyme A was discovered in the middle of last century by Fritz Lippmann, and he received the Nobel Prize for this seminal discovery. He shared this prize with Hans Krebs, who deciphered the citric acid cycle at the same time. Coenzyme A is produced in all living cells by a very conservative pathway of 5 enzymatic steps, which involve the enzymatic conjugation of ATP, pantosonate or vitamin B5, and cysteine. The presence of the ADP moiety and the sial group allows CoA to participate in numerous metabolic reactions and biochemical reactions, and to generate metabolically active CoA thioesters, such as acetyl-CoA, HMG-CoA, malonyl-CoA, and others. Now it is well known that they are critical for cellular metabolism, the regulation of gene expression, especially via protein acetylation, and the biosynthesis of neurotransmitters, cholesterol, ketone bodies, and other important cellular biomolecules. We have recently discovered a novel, unconventional function of coenzyme A, which relates to redox regulation and the antioxidant activity. It is known that exposure of cells to endogenously produced or exogenous reactive oxygen species would lead to the oxidation of surface exposed cysteines to sulfonic acid, 1-oxygen. And sulfonic acids are highly reactive and unstable. If not protected, they can be further over-oxidized to sulfinic with 2-oxygen and sulfonic state 3-oxygen. The later modification is irreversible and can lead to the inactivation and subsequent degradation of modified proteins. Low molecular weight sial, such as glutathione, it is the best known antioxidant in mammalian cells, as well as bacillosial and mycosial in bacteria, are known to bind these sulfonic acid modified cysteines and protects them from over-oxidation. These modifications are also involved in the redox signaling. Using cell-based and animal models, as well as in-house developed reagents and methodologies, we demonstrated that CoA can use this highly reactive sial group not only to generate metabolically active CoA thioesters, but to bind to oxidized cysteine when cells are exposed to stress. And this can lead to a novel post-translational modification, which we term protein coagulation. We also found, and I will discuss it later, that binding of CoA can modulate the activity, subcellular localization, and other functions of modified proteins in the antioxidant response. We started to work on CoA biology when we worked on the mTOR S6 kinase pathway, and when we searched for novel S6 kinase binding partners using the yeast to hybrid screen and affinity purification followed by mass spectrometry, we identified a novel mammalian protein. And it was about 20 years ago And this protein had extensive homology to bacterial diphospho-CoA kinase. This was quite an interesting finding for us because mTOR is known as a master regulator of cellular metabolism and biosynthetic processes in cells, while CoA is a major metabolic integrator in all living cells. Therefore, we cloned this protein and characterized and found that this protein possesses two enzymatic domains which mediate the last two steps of CoA biosynthesis in mammalian cells, and therefore we termed it CoA synthase. And here you see the biosynthetic pathway of CoA production in cells. In collaboration with Valeria Tiranti from Italy, we found that CoA synthase is mutated in patients with an aggressive form of a Parkinson-linked disorder, which is termed neurodegeneration with brain iron accumulation. Our research was focused on understanding the regulation of CoA synthase in mammalian cells initially, and we showed that it is anchored via the N-terminal hydrophobic sequence to the mitochondrial outer membrane, while both catalytic domains face the cytosol. We demonstrated that the activity of CoA synthase, or cohesive we term it, is negatively regulated via mTOR-S6 kinase and SAC kinase signaling pathway via specific phosphorylation, while signaling via stress-activated pathways would activate CoA synthase. Using proximity ligation assay and other methodologies, we demonstrated that CoA synthase function as a scaffold for the formation of the CoA biosynthetic complexes because other enzymes in the CoA biosynthetic pathway are localized in the cytoplasm. And we demonstrated that treating of cells with oxidizing agents or starving cells with serum or glucose leads to the formation of the CoA biosynthetic complex and the increase in the production of CoA, while treatment of serum starved cells with growth factors and hormones would lead to the dissociation of complex. This was quite an interesting finding, and we were really puzzled to understand why oxidative stress promotes CoA biosynthesis in mammalian cells, and we hypothesized that CoA can function as an antioxidant in protecting mammalian cells from oxidative and metabolic stress. And we indeed demonstrated the antioxidant function of CoA using several important reagents and methodologies which were developed in our laboratory and in collaboration. So, and these include highly specific anti-CoA monoclonal antibodies, which work very specifically in ELISA, Western blot and immunoprecipitation, immunofluorescence, and immunohistochemistry. And what is important to note that currently there are no anti-CoA antibodies on the market. This is not a very antigenic molecule. In collaboration with Max Gale from LMB MRC in Cambridge, we developed a reliable mass spec-based methodology for the identification of CoA-modified proteins. And to study protein correlation, we established efficient in vitro protein correlation and decorrelation assays. Now I will show you several examples of the antioxidant function and protein correlation in eukaryotic and prokaryotic cells. On this anti-CoA Western blot, you can see the treatment of red primary cardiomyocytes with a panel of oxidizing agents, hydrogen peroxide, TBH, menadiene, or diamide, induces extensive modification of cellular proteins by CoA when they are separated under non-reducing conditions. The addition of DTT would lead to the dissociation of CoA from binding proteins, and this clearly indicates that this modification is mediated via the disulfide bond. We also show that protein correlation is induced in a dose-dependent manner, and here you can see even low micromolar concentrations of hydrogen peroxide induces correlation of several proteins. Extensive protein correlation was also observed in rat hearts perfused with hydrogen peroxide, as you can see here on this slide. And you can see here, and when we probe the same lysis with anti-glutathione monoclonal antibodies, you can see strong glutathionation of modified proteins. However, the pattern of protein glutathionation and protein correlation differs significantly, indicating that these low molecular weight sials bind to different proteins or to the same proteins with different extent. We were pleased to find that protein correlation occurs not only in mammalian cells and tissues in response to oxidative and metabolic stress, but also in bacteria, yeast, Drosophila, C. elegans, virtually in all cells and organisms, model organisms. And now I will show you just one example of these studies in bacteria. Here on this slide, you can see the treatment of gram-negative or gram-positive bacteria with oxidizing agents induces strong protein correlation. And we also demonstrated that culturing bacteria in medium without glucose induces strong protein correlation, quite extensive in a time-dependent manner. So numerous proteins are correlated when cells are exposed to oxidative or metabolic stress. Why this occurs and which proteins are modified. So as I mentioned earlier, we developed the strategy, mass spec based, for the identification of correlated proteins. And this strategy allowed us to understand why protein correlation has not been identified in numerous proteome-wide screens. And in this strategy, we used anti-CoA antibodies to pull down trypsinized peptides. And then we had to remove the ADP moiety because it would lead to neutral fragmentation. And we had the specific tail of CoA, pantosine tail, bound to the peptide, which gives a unique signature. Today, we have identified, using this methodology, more than 2,000 correlated proteins. And the bioinformatic analysis revealed that majority of them in bacteria and in mammalian cells are associated with metabolic processes, about 65%. And they are involved, as you can see here, in blue and red balls, in red heart and liver of starved rats. So you can see they are involved in the Krebs cycle, amino acid metabolism, ketone body formation, beta oxidation of fatty acids, as well as glycolysis. So we identified numerous proteins and showed that the cysteine, modified cysteine, is often localized in the catalytic pocket. So correlation had the capability to modify the activity of these proteins. And indeed, we found with numerous signaling and metabolic enzymes that binding of CoA to oxidized cysteines can modulate the activity, mainly inhibit the activity, but we also found some proteins which are involved in stress response, and their activity is activated via CoA binding via allosteric mechanism. We also demonstrated that correlation can modulate subcellular localization of modified proteins, especially when cysteine, modified cysteine, is localized in close vicinity to the nuclear localization signal. Most importantly, protein correlation protects oxidized cysteine from overoxidation and irreversible damage. Today, we solved three crystal structures of covalently bound CoA to proteins and revealed that correlation can inflict significant conformational change on modified proteins. CoA is a bulky molecule. It is about 767 Daltons, and it has the ATP moiety. So we assume, but have not demonstrated, that the binding of CoA can create a novel binding platform for regulatory interactions. And the proteins which contain the Rossmann binding fold, nucleotide binding fold, can form the redox-sensitive complexes and signaling complexes. And now I will tell you why do we study NME1 as we identify more than 2,000 correlated proteins. So NME1 is the best known and the most studied metastasis suppressor protein. And this is in relationship to this meeting. We found that CoA binds NME1 via covalent and non-covalent motor interaction. This is quite interesting. This is the first protein we find to interact with CoA in two different modes. We demonstrated using recombinant NME1 that CoA binds and inhibits the activity of this protein. And we map the site of correlation, and it is located and it is specific to redox-sensitive cysteine 109. And we showed that correlation protects this cysteine from over-oxidation. How did we identify NME1? So we looked for CoA binding proteins in red tissues. You can see here liver, kidney, heart, brain, and HECT9-3 cells. And we used the affinity purification approach with CoA cephalus, CoA sulfur link, and control beads. And we were surprised to see one protein of approximately 17 kilodalton, which binds specifically to CoA agarose and CoA sulfur link. You can see here, but not to control beads. And you can see it here in HECT9-3 cells. And we were puzzled why this protein binds so specifically, and it is quite abundant. And we purified this protein and identified it by mass spectrometry as metastasis suppressor protein NME1. What is currently known about this protein? It belongs to a large family of NME proteins. There are 10 of them, and all of them possesses NDPK domain with the exception of one NME protein. NME1 is also known as non-metastatic 23 and NDPK-A. This protein is expressed at high level in tumors which have low metastatic potential. When the level of NME1 is reduced to other metastasis suppressant, these tumors are prone to metastasize much faster. What is known about the activity? So NME1 functions as a kinase, but the kinase which mediates the phosphorylation of nucleoside diphosphates. It binds the ATP, it possesses the ATP binding pocket, and transfers the ATPs via the ping-pong mechanism, it is a histidine kinase, to diphosphate nucleosides. So it replenishes the pool of NTPs. NME1 was found to function as a moonlighting enzyme because it possesses other enzymatic activities, including serine-threonine kinase, geranyl farnesyl pyrophosphate kinase activities, and exonuclease activity. So this is the major cellular metastasis suppressor protein, and the function is mediated, as I said earlier, and it was shown in numerous studies by other researchers, via the biosynthesis of GTP, CTP, and UTP in mammalian cells. The protein would regulate negatively cell motility and in cell invasion. It also inhibits cell proliferation, modulates glucose homeostasis, and apoptotic clearing. We found that it binds quite well from cells and tissues. We found the recombinant protein binds extensively to CoA beads. We also demonstrated that NME1 is correlated in vitro, and it is correlated when we overexpress transiently NME1. And on this slide, you can see that endogenous NME1 purified from HEC293 cells is efficiently correlated when cells are exposed to diamide, oxidizing agent, but not in control. And when we start cells of glucose, which is a metabolic stress, we also observe correlation of NME1. It is a monomer, and when it is oxidized, it would form the dimer. We were interested to map the site of correlation, and by mass spectrometry, we found that cysteine 109 is correlated, and it is located in a very close vicinity to histidine 118. And this histidine is a catalytic histidine which moves the phosphate from ATP, gamma phosphate of ATP, to the diphosphate nucleosides. And you can see here on this schematic diagram, this is the catalytic histidine, and cysteine 109 is located in a very close distance. Studies from other laboratories revealed that NDPK activity of NME1 is strongly inhibited by reactive oxygen species. And the metastatic function, suppression function of NME1 is dependent and is inhibited by redox-regulated cysteine 109. The structure of NME1 has been solved, NMR and crystallography, and they revealed that under physiological conditions, it would form a functional trimer with a catalytic center in the middle. However, when purified NME1 is exposed to reactive oxygen species, the trimer would dissociate, and this would lead to the formation of inactive dimer. And this dimer formation is mediated via cysteine 109, which is a redox-sensitive cysteine. So we purified recombinant NME1 and demonstrated that correlation occurs very efficiently and would significantly inhibit the activity, NDPK activity of NME1. When you add DGT and remove CoA, covalently bound to cysteine 109, the activity could be recovered. We also demonstrated that cysteine 109A mutant is not sensitive, it does not bind CoA covalently, and is not sensitive to the inhibition with CoA-CoA dimer. We were really puzzled to find that the activity of NME1 is also inhibited by non-covalent binding of CoA, just binding of CoA to the ATP binding pocket. And you can see here on this slide, my apology, that you have the inhibition of wild type NME1 in a dose-dependent manner by CoA, and as well cysteine 109 mutant is inhibited in a dose-dependent manner. So based on these findings, we proposed a model for the regulation of NME1 by CoA under oxidative stress. Under normal conditions in exponentially growing cells, ATP has much higher activity and binding affinity for the ATP binding pocket in NME1, and therefore ATP would outcompete CoA, and this would lead to the activation of the NDPK activity. However, when cells are exposed to oxidative stress, CoA would bind through the ADP moiety fast to the ATP binding pocket, and the tail would form the covalent interaction through the sial group with the redox-sensitive cysteine 109 and lock the kinase in an active state. And we call this a dual NCAM mechanism of inhibition through the ADP moiety and covalent modification by cysteine. Studies from other laboratories revealed that NME cysteine 109A mutant possesses constitutively active NDPK activity. And this mutant suppresses invasion and migration of breast cancer cells. And recently, the group in South Korea developed a small molecule activator of NME1, which binds to the C-terminus of NME1 and allosterically opens the kinase and leads to activation of this molecule. What our research is currently focused, we are interested in understanding the functional relevance of covalent and non-covalent mode of NME1-CoA interaction. We have recently solved the crystal, we have got the crystal of NME1 in complex with CoA and are focusing now to solve the structure. And our efforts are currently focused on developing novel allosteric activators of NME1 using a fragment-based approach and the crystals of NME1 with CoA. And I will finish my talk to acknowledge those people who contributed to this work, mainly Beth Yu and Maria Tostanyan from my group, as well as numerous collaborators on this project and funding bodies. I would like to thank you for your attention and would be happy to answer your questions. Thank you.
Thank you very much, Ivan, because this is an excellent talk and also mechanism of action of coenzyme A. So there is a question from Anna Dubroskova that says, is coenzyme A mediated antioxidant defense seen equally in normal and cancer cells?
No. I didn't show the results. We recently found that CoA binds much more efficiently in tissues which are in pathologies, which are associated with oxidative stress. And our efforts are mainly focused on neurodegeneration and cancer, because these pathologies are known to be associated with extensive oxidative stress. We have recently published the paper to demonstrate that protein correlation is extensively enhanced in neurodegeneration, especially in Alzheimer's disease. And we found that tau protein, which aggregates in Alzheimer's disease, is correlated in vitro and in vivo, and this correlation inhibits the dimer formation of tau. Regarding to cancer, we've got preliminary findings by immunohistochemistry that protein correlation is significantly increased in hepatocellular carcinoma and also in liver cirrhosis in patients. So this in neurodegeneration and in cancer, these are data with post-mortem brain samples and removed tumors. So clearly, this is a very good question. Yes, protein correlation is much stronger than compared to normal tissue in neurodegeneration and in cancer.
Okay, thank you. There is another question from Taras Vaglova that says, how the specificity towards single cysteine 109 residue is achieved? And also,
a brilliant question, Taras. Many thanks. CoA is a ball with ADP and a flexible tail. The tail moves always. So our current hypothesis, and we saw 3 crystal structures of CoA bound to proteins. The current hypothesis is that CoA first binds to through the ADP moiety or the pentaceine tail, and then it searches whether there is an oxidized cysteine. If there is no oxidized cysteine, it is on-off very fast. However, when it binds to the ATP moiety, and we found numerous proteins which are correlated, which contain the Rossmann binding fold. So when it binds to the protein and there is an oxidized cysteine in vicinity, the tail would lock the CoA bound through the covalent modification. So it is very different from glutathione, because glutathione is a stick of 3 amino acids with a cysteine in the middle. It's very rigid. So, and if you have catalytic pockets, very often they are quite deep. So we know from several proteins that glutathione cannot access this pocket, but CoA has a very flexible tail, so it can sneak inside of the pocket, as it is as well with NME1. Thank you.
Thank you.
Taras has also another question that says, can you exclude that in addition to the 2 lock inhibitory mechanisms proposed, an allosteric mechanism does not play a role?
It does play a role. Another good question. So we found that one of the stress activated kinases, and we did the kinase profiling screen with CoA and ADP, and found one kinase, and we published this study, Aurora kinase A, which is an important protein in the cell cycle regulation, and it is also overexpressed in numerous cancer and major target of the cancer drug discovery, that this kinase is specifically inhibited by CoA binding. So, but we also found in the kinase profiling screen of about 120 kinases that there are kinases which are activated by CoA binding. So this is not through the ATP binding pocket. And we found the cysteine is located outside of the ATP binding pocket, and this is allosteric activation through correlation.
Thank you. That's very interesting, very exciting.
We have not published this study, and it is on the shelf.
I see.
Okay.
So looking forward to solve the crystal structure of that.
And another question,